ever wondered why some people seem to age gracefully while others struggle with chronic illnesses? The answer might lie in a hidden battleground within our bodies, inflammaging. Let's delve into this fascinating area of research and uncover how inflammation and metabolism intertwine to shape our health span. Geroscience offers a revolutionary approach to tackling age-related diseases. It posits that aging and these diseases share common underlying mechanisms. These mechanisms, often referred to as pillars of aging, include factors like inflammation, epigenetic changes, and metabolic dysfunction. These pillars aren't isolated entities. They form an intricate network, influencing each other. This interconnectedness suggests that targeting fundamental aging processes could simultaneously combat multiple age-related diseases, a departure from the traditional disease-by-disease approach. Inflammaging is a chronic, low-grade inflammation that develops with age. It's sterile, meaning it's not caused by infection, but rather by the accumulation of cellular debris and other endogenous factors. This persistent inflammation plays a significant role in the development of age-related diseases. The innate immune system, with macrophages at its core, is chronically activated in inflammaging. Macrophages, first described by Metchnikoff, are key players in engulfing foreign material and initiating inflammation. However, their persistent activation in inflammaging can become detrimental. Metaflammation is metabolic inflammation driven by nutrient excess or overnutrition. It's commonly seen in metabolic diseases like obesity and type 2 diabetes. Interestingly, metaflammation shares many of the same underlying mechanisms as inflammaging, suggesting a close link between metabolism and inflammation in aging. The gut microbiota plays a crucial role in both metaflammation and inflammaging. It can release inflammatory products, influence circadian rhythms, and interact with various organs and systems. Understanding the gut's role is key to understanding these processes. Evolutionarily, organisms face two major challenges: nutrient scarcity and pathogen infection. Consequently, metabolism, immunity, and inflammation have co-evolved. Macrophages, acting as intermediaries, bridge the gap between metabolism and immunity. Macrophages and adipocytes, fat cells, exhibit functional overlap, both secreting cytokines and responding to bacterial products. The fat body in fruit flies serves as a functional unit for both metabolic and immune responses, suggesting a shared evolutionary origin. Biological degeneracy refers to the ability of different structures to perform the same function. In the context of inflammaging, a limited number of receptors, like toll-like receptors, TLRs, can recognize a wide range of signals, including self, non-self, and quasi-self molecules. These receptors, present in various cellular compartments, sense damage-associated molecular patterns, DAMPs, pathogen-associated molecular patterns, PAMPs, and metabolic products from the gut microbiota. This degeneracy, while initially beneficial for survival, can lead to chronic inflammation in aging. Identifying the stimuli that drive inflammaging is a major focus of research. Besides persistent infections, cellular debris, misplaced molecules, and misfolded proteins are major contributors. The gut microbiota, acting as a bridge between diet, metabolism, and immunity, is particularly important. A gut microbiota undergoes significant changes with age, impacting its composition and function. These changes can contribute to systemic inflammaging, creating a self-sustaining loop. Factors like age, genetics, lifestyle, and environment influence the gut microbiota's composition. Calorie restriction involving reduced calorie intake or intermittent fasting elicits cell-protective responses and extends lifespan in various organisms. It downregulates insulin signaling, mTOR pathways, and activates autophagy and stress defense mechanisms. Calorie restriction acts as a mild stressor, promoting hormetic responses. It also affects mitochondria, leading to the secretion of cytokines like FGF21 and GDF15. reducing dietary protein and amino acids is particularly effective in promoting longevity. Life-extending manipulations, like calorie restriction, often decrease core body temperature. Animals with naturally lower body temperatures tend to have longer lifespans. Calorie restriction in mammals shares similarities with hibernation, suggesting an evolutionarily conserved response to food deprivation. Hibernation involves complex neuroendocrine remodeling and a redistribution of energy. Similarities between fasting during hibernation and calorie restriction include decreased blood glucose and insulin levels. These processes are largely driven by PPR alpha. Metaflammation, or metabolic inflammation, is a low-grade chronic inflammatory state sustained by high nutrient intake. It contributes to insulin resistance and affects various organs. The ingestion of any meal, particularly those high in lipids, can trigger physiological inflammation. Postprandial lipoproteins, like chylomicron remnants and VLDL, bind to endothelial cells and leukocytes, activating inflammatory responses. They also stimulate the expression of VCAM1, promoting monocyte adhesion. During nutrient overload, the innate immune system is excessively stimulated, leading to homeostatic failure and metabolic disorders. High-fat meals can increase serum levels of bacterial endotoxins, lipopolysaccharide, causing leukocyte activation and inflammation. Bacterial endotoxins are potent inflammatory compounds that circulate at low concentrations in the blood, mimicking a chronic low-grade bacterial infection. A high-fat diet can cause gut microbiota dysbiosis, exaggerating lipopolysaccharide production. High-fat diet. Adipose tissue plays a central role in metaflammation. Adipocytes exposed to nutrient-dense diets increase in size, leading to reduced vascularization and hypoxic conditions. They also experience endoplasmic reticulum (ER) stress, activating inflammatory pathways. ER stress triggers inflammation through the unfolded protein response. This response involves the activation of mTOR and downregulation of AMPK and SIRT pathways. Inflammaging and metaflammation, while originating from different research fields, share common features. Both involve chronic, sterile inflammatory processes that contribute to metabolic diseases and age-related decline. The mechanisms sustaining chronic inflammation are largely shared. These mechanisms include increased senescent cells, hyperactivation of the innate immune response, and accumulation of cellular debris. Metaflammation can contribute to inflammaging and be considered a nutrient excess-driven accelerated aging. Metaflammation might precede and contribute to inflammaging, and vice versa. Metabolic age-related diseases could be considered manifestations of accelerated aging. This suggests that an individual's metabolic history affects their immunobiography and inflammaging phenotype. This view opens the possibility of using aging biomarkers to support the diagnosis of metabolic diseases. These biomarkers should be capable of tracking healthy and unhealthy metabolic aging. Examples include DNA methylation, glycomics, metabolomics, and lipidomics. DNA methylation, an epigenetic modification, can distinguish chronological age from biological age. Mathematical models, termed epigenetic clocks, are based on DNA methylation levels at multiple CpG sites. These clocks can estimate an individual's biological age and predict health outcomes. The Horvath's epigenetic clock, based on 353 CpG sites, has been applied to various conditions. It shows age acceleration in Down syndrome and Werner syndrome, and decelerated aging in centenarians. Glycosylation, a post-translational modification of proteins, modulates various biological functions. Analyzing sugar chains attached to proteins, N-glycans, can identify new biomarkers of aging. N-glycans devoid of galactose residues are potential biomarkers. Enzymatic glycosylation has become a promising biomarker of biological age. The glyco-age pest, based on the ratio of two N-linked glycan species, increases with age and is associated with healthy and unhealthy aging. Metabolomics and lipidomics can provide biomarkers at the interface between metabolism, inflammation, and disease risk. High-throughput technologies allow the measurement of a wide range of endogenous small molecule metabolites. Lipidomics can be a source of biomarkers of biological age and longevity. Centenarians have a unique lipid profile with changes in specific lipid species. Metabolomic profiling has also been used to identify biomarkers of metabolic diseases. The nutrient composition, quantity, and timing of meals have a strong effect on the gut microbiota and metabolism, contributing to a basal level of inflammation. Metaflammation, caused by nutrient excess, can be considered a specific case of a more general inflammatory mechanism. The integration of inflammaging and metaflammation within the geroscience perspective suggests that metaflammation can be considered a specific type of accelerated aging. This paves the way for the amalgamation of biomarkers and improved preventive and personalized medicine for elderly individuals. Ready to take control of your health span? Start by exploring the pillars of aging and how they relate to your lifestyle. Consider adopting a balanced diet, engaging in regular physical activity, and consulting with healthcare professionals to assess your biological age and develop a personalized plan for healthy aging. Share this knowledge with your friends and family, and let's work together to unlock the secrets of longevity.